air injection into veins. Do you know who this man is? This is General Shiro Ishii, and he had a deep interest in the human body, more specifically in how it failed, like how the human body reacts to disease, what happens when we are exposed to extreme heat or cold, and even what happens if we shuffle our organs and limbs places. To get those answers, he built one of the most horrific research facilities in human history. In 1936, the Imperial Japanese Army officially set up a secret complex in Harbin, China, officially known as Unit 731, and it was run by Ishii himself. The goal was simple: build biological weapons for war. And to do that, they used thousands of Chinese, Korean, and Soviet civilians and prisoners of war as guinea pigs. The subjects were called logs, so the staff could pretend they were not human. But before we get into all those messy tests, one of their most brutal ideas didn't even need a fancy disease or gore experiments. They just needed a syringe. For the human body, even a small air bubble in your vein is dangerous because it could travel toward the heart or lungs and starts blocking your blood flow. And if enough of it builds up, the heart basically gets stuck. It can't push blood forward anymore. So the brain and every other organ just stops getting oxygen, and the person can die in minutes. And that's exactly why the doctors at Unit 731 paid attention to it. If air could shut down the human body that fast by accident, they wanted to see what would happen if they controlled it on purpose. The thought Shiro Ishii and his team had was, just how much air is enough air? So, inside the facility, the doctors pushed controlled amounts of air into prisoners' veins using syringes. They noted the volume injected, then watched for the first reaction. Within minutes, the prisoners started struggling to breathe, clutching their chest as the air bubble traversed through the heart and blocked circulation. And as the blood flow failed, the body just weakened and folded. The doctors tracked how long it took from injection to loss of consciousness and then heart attack arrest. From their experiments, today we know it only takes about 200 to 300 milliliters of air to kill someone. That is just roughly 1 cup. Just 1 cup of air pushed fast into a vein, and the heart is done. Repeated blood withdrawal. In the late 1930s, Japan's war in China was dragging on, and they had a consistent problem. Soldiers kept dying, but not always right on the battlefield. A lot of them bled out before anyone could stabilize them. Military doctors needed to know exactly how much blood a person could lose before their body gave up, and they had a building full of prisoners they could use to find out. The process was simple. They would pick a prisoner, strap them to a table, and slide a needle into their vein. Then they started pulling blood out. Not all at once, but in stages. About 500 milliliters at a time, roughly a pint every 2 to 3 days. And they just kept going, draw after draw, watching exactly what changed in the body each time. As the blood volume dropped, the heart would start beating faster because it was trying to push whatever blood was left. The body would pull blood away from the skin and the limbs, then send it all toward the brain, the heart, and the kidneys. As more blood came out, blood pressure would fall, and the brain, getting less and less oxygen, would start going foggy, dizzy, until the person could barely stay conscious. The human body can adjust to small losses, but once the blood loss crosses a critical level, around 40% of your total blood volume, the heart can't supply oxygen to vital organs, and organ systems begin shutting down one by one until the heart gives out completely. That stage is called hemorrhagic shock. And no, they didn't stop once the prisoner died. Doctors cut the body open immediately and examined everything inside. The heart would be swollen from overworking itself trying to pump blood that wasn't there anymore. The kidneys would be shrunken and damaged because the body had been starving them of blood flow to keep the brain alive. The gut would be wrecked because it was one of the first things the body sacrificed when blood got low. And the brain, the organ that needed oxygen the most, would show signs of cell death from being slowly suffocated over days. Animal blood injection. Now that the doctors at Unit 731 know how much blood a person could lose before dying, they move to the other side of the problem: how to put it back. Because out on the battlefield, soldiers were bleeding out faster than medics could help them. Human blood wasn't easy to store and hard to deliver. UPS guys can't travel well through war zones, so the staff started asking a different question. What if they didn't need human blood at all? What if they could use animals instead? Horses were everywhere in the 1940s military. Every camp had them, and a single horse carries way more blood than a human does. So if they could pump horse blood into a wounded soldier and have it actually work, it would basically have a walking blood bank on every front line. But they already knew that animal blood isn't compatible with the human body. As far back as the 1600s, doctors tried transfusing lamb and calf blood into people, and a lot of them died. By the early 1900s, scientists found that the human immune system treats animal blood cells as invaders and immediately tries to destroy them. But knowing that something is dangerous and knowing exactly how it kills you are two different things. The doctors at Unit 731 wanted to watch the entire process from start to finish in a controlled setting, measuring every stage of the breakdown, and see if there was any volume or method that might somehow work. So they picked healthy prisoners and pushed large amounts of horse blood directly into their veins, and the body freaked out almost immediately. Their immune system flagged it as a threat and started destroying it. But when your body destroys that many foreign cells at once, all the gunk inside those cells spills out into your bloodstream like trash being dumped into a river. That trash gets pushed to the kidneys, which are basically your body's filters, and they get so overloaded they start shutting down. Meanwhile, the rest of the body is panicking. Fever shoots up, blood pressure drops, and the lungs start filling with fluid so the prisoner can barely breathe. So the body is literally fighting itself from the inside out. Like every other experiment, the doctors moved the body to a table right away and opened the chest. The kidneys were usually the most visibly destroyed, swollen and dark from trying to filter all the trash. The major blood vessels had thick, dark clots sitting inside them, and the lungs showed signs of fluid buildup from the inflammatory response that had overwhelmed the body in its final minutes. Seawater injection. So, horse blood was a disaster, and every prisoner who got it died. But the core problem hadn't changed. Soldiers were still bleeding out on islands across the Pacific, and there was never enough blood or medical supplies to go around. The staff needed a backup plan, something that could keep a wounded soldier alive even when real blood wasn't available. In the medical world, usually doctors use saline to keep someone alive when they lose a lot of blood. It's just clean water with salt mixed to match the fluid inside human blood. When it enters your veins, it gives your heart a bit more volume to pump so you don't die right away. It only buys time, but it keeps you alive long enough for real blood to arrive. But hauling bags of saline across islands in the Pacific wasn't easy. Seawater, on the other hand, was everywhere and free. So doctors wondered if they could skip the medical supplies and replace saline with whatever they scooped out of the ocean. To test this idea, the doctors grabbed healthy prisoners and pumped raw seawater right into their veins. Now, what happened was, each blood cell is like a soft little bag filled with water, and salt controls how much water stays inside that bag. If the salt level outside the cell matches what's inside, everything's fine. But seawater has roughly 4 times more salt than your blood does, and salt always pulls water toward it. So the moment that seawater hit the bloodstream, it started sucking the water right out of every blood cell it touched, collapsing them like tiny deflated balloons that could no longer carry oxygen. The water rushed out so fast that the cells collapsed and shriveled into small dried up balls that couldn't carry oxygen anymore. Once enough cells collapsed, the blood turned thick and heavy, and the heart had to fight to push it forward. That struggle threw the whole body off balance. The brain couldn't get normal oxygen or fluids anymore, so pressure started building inside the skull. As everything slipped out of control, the prisoners began shaking, lost awareness, and finally collapsed when their organs couldn't keep going. After that, the staff moved the bodies to a table right away for their observation. The kidneys were swollen and pale because all that extra salt overwhelmed them. The blood inside the veins looked thick and grainy, and across every organ, they looked the same. It had been drained of water and shriveled up. In the end, the doctors knew seawater killed everyone who received it, but they kept running the tests over and over anyway, because they weren't trying to save anyone at that point. They just wanted a complete set of data for their files. Syphilis transmission. In the early 1940s, the Japanese army had a problem bigger than any weapon. Japanese soldiers in occupied territories were assaulting local women and using pleasure slaves. And because of that, syphilis was spreading in their ranks like wildfire. The infected soldiers could lose their strength, their eyesight, and even their ability to think straight. Basically, Japan was losing fighting power not to bullets, but to an STD. Their commanders needed to understand how the infection actually moved through the body, and how fast it destroyed a person from the inside. So the doctors at Unit 731 did what they always did. They turned to the prisoners. The goal was to watch syphilis from the exact second it entered a person all the way to the final stages. At first, they tried injecting the bacteria directly into the prisoners, but that didn't work well, because when the bacteria went straight to the blood, it skips the natural entry point and makes the infection develop differently than it would in real life. The doctors didn't want a shortcut. They wanted to know how the disease actually spreads the way soldiers were catching it. So they switched to something far worse. They took male prisoners who were already infected with syphilis and forced them to assault uninfected prisoners, both women and men, while guards watched and recorded the exact time it happened. This gave doctors the exact moment the disease hopped bodies through natural transmission. After the assault, the staff checked the victims every day. First, a sore would show up, then fever, then swelling. And finally, rashes started popping across the skin as the bacteria worked its way deeper into the blood. Eventually, it hits the nervous system, and that is when the things get really bad. Because once syphilis reaches the brain and spinal cord, the person starts losing control of their own body. Once the infection hit that point, they strapped the prisoners to a table while they were still alive, cut them open without any painkillers, and examined the liver, the heart, and the major blood vessels to see exactly how far the disease had eaten into them. And they didn't stop at adults. A youth corps member who trained at the facility remembered seeing the people lined up for syphilis testing one day. A Chinese woman holding a baby, a Russian woman with a little girl around 4 or 5, and another Russian woman with a boy who looked about 6 or 7. The doctors put those children through the same tests as their mothers. On top of that, the staff forced female prisoners to get pregnant on purpose because they wanted to know if a mother with syphilis could pass it to her baby before it was even born. Once the pregnancy was far enough along, they cut the mother open alive and pulled the fetus out to check. A lot of babies were born inside that facility over the years. Not a single one made it out. Each stage gave them another piece of the infection timeline, and they kept collecting those notes as the war dragged on. Cholera infection. Imagine prisoners locked inside a small cell in Unit 731. They haven't had clean water for a full day. Then, a guard walks in and hands them cups of clear drinking water. The men drink fast because they're dehydrated. But what they don't know is that the doctors had mixed the water with cholera bacteria. In the 1940s, the Japanese army saw how devastating cholera were in the field and wanted to turn them into weapons. The idea was simple. If you could drop bacteria into an enemy's water supply, you could wipe out entire towns without bullets. But there was a catch. If your own soldiers walk into that same town water, they'll be drinking the same water. So the army needed two things: a reliable way to spread the disease and a vaccine to keep their own troops safe. And that's what the doctors at Unit 731 set out to build. They used the prisoners and split them into two groups. They gave the first group a vaccine shot and left the second group with nothing. Then every prisoner drank contaminated water or ate contaminated food. Now here's what these diseases actually do to you. Cholera goes after your gut and basically flips a switch that makes your intestines dump all your body's water out at once. So you're hit with non-stop diarrhea and vomiting so bad that you can lose liters of fluid in hours. Your body dries out way faster than you can drink anything back. And this disease hit those prisoners hard. The unvaccinated group fell apart fast. They lost so much water non-stop, you could actually see their skin started shrinking around their bones. Their eyes sank in, and they got so weak they couldn't even stand up anymore. As the sickness got worse, the staff didn't wait around for anyone to die on their own. They picked victims at different stages and cut the stomach open while the organs were still working. They observed the victim's stomach lining, intestines, and the fluid in the cavity to see how far the disease had spread. And then they compared it with the vaccinated group to see if the vaccine actually slowed any of the damage down. Nobody walked out of this. If someone died from the sickness, the doctors just dissected them. If someone survived because the vaccine worked, congratulations, you get dissected too. The staff kept both sets of organs so they could write complete reports. But the experiments inside the compound were only half of it. Unit 731 also took their bacteria out into the real world and tested it on entire cities. Japanese planes flew low over Chinese villages and dropped infected food, contaminated clothing, and even poisoned candy onto unsuspecting civilians. They dumped cholera into wells and water supplies across at least 11 Chinese cities between 1940 and 1943. The resulting outbreaks killed an estimated 200,000 to 400,000 civilians. By 1945, as Japan faced defeat, the staff burned any evidence of what happened. But when investigators reviewed what survived after the war, they found that many of the vaccine batches barely worked at all. The doctors had repeated the same fatal tests over and over because they couldn't reach the protection level the army wanted. Thousands of people died for a weapon that never became reliable, and a vaccine that never actually worked. Weapon testing. Imagine a wide dirt field with wooden poles planted in a circle with prisoners tied to them. Some were placed right beside a small dirt pit in the middle of the field, while others were spaced out at distances of around 3 meters, 6 meters, or even 10 meters from it. Then, suddenly, a soldier drops a live grenade into the center hole and dives into a trench, while the prisoners just hopelessly wait for the blast and flying bits to rip them to pieces. But grenades were just the start. The military wanted to know exactly how every weapon in their arsenal performed against a real human body. So they turned that field into a full-blown shooting range. They lined prisoners up and fired at them with Nambu 8mm pistols, bolt-action rifles, and machine guns from different distances to compare wound patterns and how deep the bullets went. They tested flamethrowers on both covered and exposed skin. They even had prisoners tied down while soldiers stabbed them with bayonets, swords, and knives so the doctors could study blade wounds. Basically, if the Japanese army owned it, they tested it on a living person. Right after each test, the medical staff rushed toward the wounded prisoners, but not to save them, but to document what happened. They checked which organ got hit, how deep the fragments or bullets went, and how long a wounded person could stay alive before bleeding out. They wrote down the time to collapse and the time to death for each distance. And for the ones who survived the initial blast or shot, the doctors had a second experiment running. They left the fragments inside the body on purpose. It's because shrapnel carries dirt, rust, clothing fibers, and bacteria deep into the wound. And the doctors wanted to know exactly how long it took for that contamination to turn into an infection and kill the person. Over the next few days, the staff watched the wounds swell, turn dark, and leak as the infection. They wanted to see if a single explosion could kill twice, once with the blast and again with infection. By the end of the war, the charts and sketches from these field tests stayed hidden inside military files. Years later, when investigators finally reviewed them, they noticed the blast patterns were inconsistent. Different soil types changed the fragment spread. Weather changed the kill range. Even the same grenade type didn't behave the same way twice. After all the bodies and all the tests, the data wasn't reliable enough to predict real battlefield results. The people tied to the poles died for numbers that didn't even work outside that field. Frostbite exposure. In the 1940s, the Japanese army was preparing for a winter war against the Soviet Union. They knew that in extreme cold, a soldier's hands and feet could freeze solid. To prepare for this, one of the lead researchers at Unit 731, Hisato Yoshimura, decided to use prisoners to map out exactly how long it takes for human flesh to turn into ice. To do this, Yoshimura's team waited for the coldest nights in Manchuria and turned the outdoor yard into a freezing lab. They lined prisoners up, made them dip their bare hands and feet into tubs of ice water, and then forced them to stand in the open wind with wet skin. This way, the cold hit harder and faster. To tell if the experiment was successful, the doctors used a simple test. A researcher would walk up to a prisoner and strike their frozen arm or leg with a short wooden stick. If the limb was frozen all the way through, it would make a hard clacking sound, like hitting a piece of dry wood or a stone. The researchers kept the prisoners in the wind until every single person made that same sound. Once the limbs froze, the team dragged the victims indoors for the next stage. Yoshimura wanted to see how frozen skin reacts at different temperatures, so the doctors lowered the limbs into buckets that ranged from lukewarm to almost boiling. As the water hit the skin, the damage showed itself fast. Lukewarm water made the frozen flesh swell and split open, while the hotter water broke the tissue so severely that skin and muscle peeled away from the bone. After Japan surrendered in 1945, Yoshimura walked away free. The United States gave him immunity in exchange for his data, and he went home to build one of the most respected medical careers in post-war Japan. He got appointed principal of Kyoto Imperial Medical University, founded the Japanese Society of Biometeorology, and published papers in hypothermia and cold resistance in academic journals throughout the 1950s. But none of those papers mentioned that the data came from torturing prisoners. Other researchers around the world cited his work for decades, treating it as legitimate science, because nobody in the West even believed Japan had run human experiments until the 1980s. Everyone just assumed it was Soviet propaganda. At 77, when a reporter from the Mainichi Shimbun newspaper confronted him about the experiments, Yoshimura just shrugged it off and said it was the country's fault, not his. His victims never received names or graves, while Yoshimura got a career, a legacy, and a long, comfortable life. Heat dehydration. Unit 731 had a room specifically designed to turn a living person into a mummy. Not metaphorically, literally. A sealed, windowless room with industrial fans mounted inside, built for one purpose: to pull every drop of water out of a living human body. Now, why would anyone want to do that? Well, scientists back then had known that the human body is mostly water, but all of that research came from studying corpses, and Unit 731 didn't trust dead body data. They figured a living body might hold water differently than a dead one, and the only way to find out for sure was to dry a living person out completely and weigh what was left. They also wanted to know how long the body could last during the process, how fast the organs would fail, and at what point there was no coming back. And since nobody was going to volunteer to be slowly dried to death, they used prisoners. Here's how it worked. First, they starved the prisoners before the experiment even started. Then they dragged them into a sealed, windowless room, sat them down, and turned on industrial fans that blasted hot, dry air directly at their bodies, basically turning the room into a giant hairdryer that you can't escape from. Now, here's what happens when you point that kind of heat at a living person. The body immediately starts sweating to cool itself down, but the hot, dry air evaporates that sweat the moment it hits the skin. So the body panics and pushes out even more sweat, and the fans evaporate that too. This loop just keeps running, and the body loses water faster and faster with no way to replace it. And once that water is gone, everything goes wrong at once. Blood is mostly water, so when the body dries out, the blood gets thicker and thicker until it turns into something closer to sludge. Now, the heart is trying to pump that sludge through your veins, but it's harder than before, which means your organs stop getting enough oxygen. Your kidneys start shutting down, your skin pulls tight against the bones, and eventually your heart just gives out because it can't push anymore. When it ended, the researchers dragged the body onto a scale. The corpse weighed roughly 1/5 of what that person weighed when they walked in. That means about 78% of their entire body had been water, and the fans had pulled every drop of it out. Japanese writer Seiichi Morimura documented this in his book The Devil's Gluttony, based on interviews with Unit 731 veterans. The doctor's conclusion? The human body holds an enormous amount of water, and without it, a person dies faster than almost anyone had expected. Limb reattachment. Imagine you wake up from a deep sleep in a quiet white room. You look down and something is wrong. Your left hand is now on your right side, and your right arm is on your left shoulder. Someone swapped your limbs while you were unconscious. That actually happened at Unit 731, and the reason behind it is somehow even worse than it sounds. Soldiers across Asia were losing arms and legs to barbs and bayonets every day, and military commanders wanted to know if a severed limb could be reattached and still work. That's at least a question with purpose, but the surgeons at Unit 731 took it way further than that. They started asking things like, what if you put the right arm on the left side? What if you swap both legs? What happens to the nerves, the blood flow, the function? And based on what historians have found, a lot of these experiments had no real military goal at all. The researchers just wanted to see what would happen, like a kid pulling the legs off an insect, except the insect is a living human being. So the surgeons got to work. They put prisoners under, amputated both arms or both legs, switched them, and stitched them onto the wrong sides, right onto left, left onto right. But here's a problem. A right arm and a left arm aren't interchangeable. The blood vessels, nerves, and bone structure on each side sit in mirrored positions. So when the surgeons tried to attach a right arm to a left shoulder, nothing lined up. They had to physically twist the limb to force the vessels to connect, which meant the hand ended up facing backwards with the palm pointing the wrong way. And then they just stitched it shut and waited to see what would happen. Within hours, the swapped limbs started changing color. First, they went pale, because blood couldn't get through the twisted connections. Then they turned purple, as blood without oxygen pooled with nowhere to go. And eventually, the whole limb went black, which means the tissue is dead. Once that starts, it doesn't stop. The limbs swelled up, blisters broke through the skin, and bacteria moved in fast, because dead tissue on a warm body is basically an all-you-can-eat buffet. Every single reattachment failed. Not one survived. None of this ever helped a single Japanese soldier. Many historians have described these experiments as utterly pointless, but the surgeons who ran them never saw a courtroom. Vivisection on conscious subjects. Imagine you've had a deadly fever for 3 days straight because the guards forced you to eat infected food. Then, a group of men in full rubber suits and gas masks drag you into a sterile room and strap you down on a cold steel table. You think the doctors are finally here to treat you, but the doctor doesn't reach for any medicine. He picks up a scalpel, hands it to a student, and points at your stomach. Before you can say anything, the blade is already pressing into your skin. This was actually a common practice at Unit 731. For context, Unit 731 existed to study how diseases destroy the human body. And to do that, they needed prisoners who were already sick. So, weeks before any surgery, researchers injected prisoners with cholera, typhoid, or plague, or forced them to eat food laced with bacteria. Then they sat back and watched, waiting for the infection to hit its absolute worst point. Once it did, they needed to see exactly what the disease was doing to the organs. Now, the obvious approach would be to wait for the person to die and then open them up. But here's the problem. The moment a heart stops, the body starts to rot. And that rot contaminates the organs and ruins the data. Everything is damaged, and you can't tell what broke first. So the only way to get a clean look at how the disease spread was to cut the person open while they were still alive. So, at the peak of infection, guards dragged the prisoners out of their cells and strapped them down by the wrists and ankles. And there were no painkillers, because the doctors believed the chemicals would change how the bacteria behaved inside the body. So the prisoner felt every single cut. If they screamed, the doctors kept working. If the screaming got too loud, they just stuffed a rag in their mouth. Then the lead doctor sliced the torso open from the chest all the way down to the lower stomach, pulled out the lungs, liver, and kidneys one by one, and examined them while the person was still conscious. The whole thing only ended when the prisoner bled out and died on the table. But disease research wasn't the only reason they cut people open alive. The Japanese army also used vivisection as a training program for new military surgeons. These were doctors who had never operated on a real living body before. And instead of practicing on cadavers like normal medical schools, the army handed them prisoners. After the war, one of these surgeons actually came forward. Army Dr. Ken Yuasa admitted that when he performed these vivisections, they were, in his words, all for practice rather than for research, and that this kind of thing was routine across mainland China, with at least 1,000 Japanese military personnel taking part. Some historians have even suggested that the real goal wasn't to build surgical skill at all, but to desensitize the doctors so they wouldn't hesitate when they got to the battlefield. Over 3,000 people died this way, and that's the most conservative estimate. Former Unit 731 member Okawa Fukumatsu claimed that the real number was closer to 10,000, and other estimates range as high as 12,000. When the war ended in 1945, the American government struck a deal with General Shiro Ishii. Every single researcher got full immunity from war crimes prosecution. No trials, no prison. The Americans got all the research data in exchange. Experiments on pregnant women. In the entire history of Unit 731, not a single baby born inside that camp survived the war. Every single one ended up in a glass jar or an incinerator, and the women carrying them weren't spared either. The doctors wanted to know how diseases and weapons affect a developing baby inside the womb. Things like, can syphilis pass from a mother to her unborn child? What happens to a fetus when you inject the mother with plague? Does a chemical weapon kill the baby first or the mother first? And the only way to study any of that was to get pregnant women into the camp and run the experiments directly on them. But captured pregnant women were rare, so they created their own supply. Guards dragged male prisoners into female cells and forced them to assault the women. And they didn't pick these men at random. They specifically chose ones who were already infected with diseases like syphilis, because that way the assault doubled as a disease transmission experiment. The guards watched the whole thing to make sure it happened, and once a pregnancy was confirmed, the experiment started. From that point on, the woman's body became a lab. At times, they injected her with anthrax or plague. They tracked whether the bacteria crossed the placenta and reached the baby before it killed the mother. And if they were testing weapons, it got even worse. The doctors locked pregnant women inside chambers and flooded them with chemical agents like mustard gas and phosgene, which are chemicals that burn your skin, destroy your lungs, and eat through soft tissue. Then they watched what happened. Did the gas reach the baby through the mother's blood? Did the fetus die before the mother did? Or did it keep developing with damage? How far along did the pregnancy need to be before the chemical affected the baby differently than it affected her? And chemical weapons were just one category. They hit pregnant women with shrapnel from controlled explosions to see how blast fragments traveled through a pregnant body compared to a non-pregnant one. Shot them at different ranges to study wound depth near the uterus, and crushed parts of their bodies to measure how blunt force trauma affected the fetus at different stages. Every type of weapon the Japanese army had, the doctors tested on a pregnant woman at some point. And that meant they needed pregnant women at every stage. First trimester, second trimester, third trimester, all at the same time, which is why they kept forcing prisoners to get women pregnant over and over again. Then came the operating table. The doctors strapped the mother down and cut her open while she was still conscious with no painkillers. Because just like every other vivisection at Unit 731, they believed anesthesia would contaminate the results. One account from a doctor at the facility described a pregnant woman waking up mid-surgery who said, "It's all right to kill me, but please spare my child's life." Then they pulled the fetus out and examined it. Every experiment ended the same way. Both of them died on the table.